During oxidative phosphorylation, chemiosmosis couples electron transport to ATP synthesis. Following glycolysis and the citric acid cycle, NADH and FADH2 account for most of the energy extracted from food. So high energy electron gonna get trapped into NADH and FADH2, and these NADH and FADH2, that electron carrier, they donate electrons to the electron transport chain, which powers ATP synthesis via oxidative phosphorylation. The pathway of electron transport. The electron transport chain is in the inner membrane we call crista or cristae of the mitochondrion. Most of the chain's components are protein in electron transport chain, which exist in multi-protein complexes. The carriers alternate reduced and oxidized states as they accept and donate the electron. Electrons drop in free energy as they go down the chain and are finally passed to O2, forming H2O water. The final stage of cellular respiration is oxidative phosphorylation through electron transport chain to produce ATP. That's the major mechanism to produce most of ATP in cellular respiration. The diagram explained about electron transport chain at inner membrane of mitochondrion. When NADH, the electron delivery man, or FADH2 donate electron to electron transport chain, the donated electron, they're gonna pass through one after another, each carrier protein, all the way go into the last and final stage. They drop electron to O2 because O2 is waiting for electron to drop down, because this O2, one of the strongest electron acceptor, pulling down the electron to their side. And after getting electron, they turn into H2O. And each carrier molecule, after they accepting electron, they reduced and giving to the next. After giving to the next, donate to the next electron, then it's gonna become oxidized. So each carrier molecule, after they getting electron, they reduced, and after giving to the next, they oxidized. And next one, when they receiving electron, they reduced and giving up and oxidized. So reduced, oxidized, reduced, oxidized. So every carrier molecule, after they getting electron, they reduced, and they giving up to the next, they oxidize. So they change, reduced, oxidized, reduced, oxidized. They repeating that and giving the electron, finally giving to O2. So that's the electron flow, yeah. That's why we call that's the electron transport chain all the way into O2 at inner membrane of mitochondrion. Electrons are transferred from NADH or FADH2 to the electron transport chain. Electrons are passed through a number of proteins, including cytochrome. That means the containing iron. So each with an iron atom, 2O2. The electron transport chain generate no ATP directly, but they produce indirectly. It breaks the large free energy drop from 2 to O2 into smaller steps that release the energy in manageable amounts. When electrons go through the electron transport chain all the way into O2, the electron pass through every carrier molecule. And that carrier molecule, they change the form into reduced form and oxidized form, reduced form, oxidized form, reduced form, oxidized form, all the way into O2. And for the reduced form, sometimes they accept electron, but at the same time, they accept H plus ion. And when they oxidize after giving up electron, sometimes they losing electron to the next, and sometimes they losing H plus ion, proton to some area. So giving up to the next one, or they giving into the environment. So when they change reduced form and oxidized form, reduced form, oxidized form, passing through the electron, the result, whenever they flow the electron through the electron transport chain, they have an effect to pump H plus ion, pump proton through that membrane. So when electron go through that electron transport chain, it can cause H plus ion pump, proton pump from a matrix through the inner membrane into intermembrane space. One more time. So electron transfer in the electron transport chain causes proteins to pump H plus ion from the mitochondrial matrix, the internal space, into the intermembrane space through that inner membrane. H plus ion then moves back across the membrane, passing through that protein complex, ATP synthase. So that's why when electrons go through that electron transport chain, they make pumping of H plus ion into intermembrane space. That's why inside the intermembrane space, they have high concentration of H plus ion. When they make a high pressure, they have to go back, otherwise it's gonna explode. When they move back into the matrix, they using the tunnel provided by the protein complex, so-called ATP synthase. That's why ATP synthase uses the flow of H plus ion, hexagonic flow of H plus ion, never mind hexagonic, but so high pressured H plus ion is gonna release out through that ATP synthase. And those, flowing power to drive phosphorylation of ATP. So using that flowing out power of H plus ion through the ATP synthase, it can be used up for phosphorylation of ADP into ATP. So this is an example of chemiosmosis, the use of energy in a H plus ion gradient to drive cellular work to produce ATP. This picture showing the oxidative phosphorylation. When NADH and FADH, electron delivery man, deliver the electron to electron transport chain, then electron go flow through that electron transport chain. So when electron go through that electron transport chain, it has an effect to pump H plus ion from matrix into intermembrane space. So when many NADH and FADH, they donate many electrons. When many electrons go through the electron transport chain, then H plus ion concentration gonna increase up very sharply. So the pressure of H plus ion gonna get released back through that ATP synthase. As you can guess, this protein complex enzyme, ATP synthase, they supposed to produce ATP. So they using the H plus ion flowing through that ATP synthase enzyme complex using the flowing power of H plus ion, we call H plus ion gradient. So they catalyze the site for phosphorylation of ADP into ATP. So they produce ATP by chemiosmosis. So when we regard H plus ion as a chemical, then it's gonna diffuse out back. So we call chemiosmosis. So using the chemiosmosis, they produce ATP. That's the what? That's the oxidative phosphorylation. So most of the ATP in cellular respiration, they produced by oxidative phosphorylation. So as we already discussed over, this oxidative phosphorylation, that's the major mechanism for the production of ATP, mass production of ATP in cellular respiration, in addition to substrate level phosphorylation in glycolysis and Krebs cycle. So they're using H plus ion pressure, H plus ion concentration gradient. So the energy stored in a H plus ion gradient across a membrane couples the redox reaction of the electron transport chain to ATP synthesis. The H plus ion gradient is referred to as a proton motive force, emphasizing its capacity to do work. So they're using proton motive force to produce ATP by ATP synthase. Accounting of ATP production by cellular respiration. During cellular respiration, most energy flows in the sequence: glucose, NADH, electron transport chain, proton motive force into ATP. The energy in a glucose molecule is transferred to ATP during cellular respiration, making about 32 ATP. It depends on biology textbook. Some biology textbook, they said from one molecule of glucose, they can produce 36 to 38 ATPs, or the other textbook, they say about 30 to 32 ATP can be produced from one molecule of glucose by cellular respiration, because there are some controversy for the productivity of ATP through cellular respiration from a glucose molecule. So there are several reasons why the number of ATP is not known exactly, but we're not going to discuss about that for now. This one, overall summary for cellular respiration. So it is composed of glycolysis and citric acid cycle and oxidative phosphorylation through electron transport chain. For the glycolysis, they produce 2 ATP by substrate level phosphorylation, but they reduce 2 molecules of NADH. And just before the citric acid cycle, they reduce 2 molecules of NADH, and through citric acid cycle, they produce 2 molecules of ATP by substrate level phosphorylation, but they reduce 6 molecules of NADH and 2 molecules of FADH2. So those all, the total for the NADH from 1 molecule of glucose, that's about 10 molecules of NADH and 2 molecules of FADH2. They deliver the electron into electron transport chain. Then from that 10 molecules of NADH and 2 molecules of FADH2, they produce about 26 or 28 ATP molecule by oxidative phosphorylation. So total is about 30 or 32 ATP molecule produced from 1 molecule of glucose. Those NADH produced from glycolysis, because glycolysis occurs in cytosol outside the mitochondrion. That's why when they use NADH from that glycolysis, they have to go through the membrane into the mitochondrion. Sometimes those NADH produced outside, when they go through the membrane, they have to take active transport. When they go through the active transport shuttle, then they have to pay ATP. They have to consume ATP. That's why the overall productivity, it can be reduced down sometimes. Sometimes when it is lucky, they can go through the mitochondrial membrane freely sometimes. Depending upon the shuttle system, it can affect the final productivity for ATP, about 30 or 32 ATP produced from 1 molecule of glucose. Fermentation enables cells to produce ATP without the use of oxygen. Most cellular respiration requires O2 to produce ATP. Without O2, the electron transport chain will cease to operate. So, without O2, the electron transport chain will shut down, cannot work. In that case, glycolysis couples with fermentation to produce ATP. We already discussed over the substrate level phosphorylation. It occurs in glycolysis. So fermentation uses substrate level phosphorylation instead of an electron transport chain to generate ATP. Actually, we already saw that 2 ATP produced by substrate level phosphorylation in glycolysis. Actually, that's all productivity for fermentation. That's all. The most common types of fermentation, we can see two. One is alcohol fermentation, and the other one is lactic acid fermentation. So types of fermentation. Fermentation consists of glycolysis plus reactions that generate NAD+, which can be reused by glycolysis. In some steps of glycolysis, they need NAD+ for the acceptor for electron after glucose broken down. So when glucose broken down, electron come out, that electron can be accepted by NAD+. That's why NAD+ is required for the glycolysis going on. So there are two common types of alcohol fermentation and lactic acid fermentation, as I already discussed over. In alcohol fermentation, pyruvic acid is converted to ethanol, ethyl alcohol, in two steps. The first step releases CO2, carbon dioxide, and the second step produces ethanol, ethyl alcohol. Alcohol fermentation by yeast is used to brewing, winemaking, and baking. This one showing you two types of fermentation: alcohol fermentation and lactic acid fermentation. Their end products are, first one is alcohol and second one is lactic acid. That's why we call by their end product alcohol fermentation or lactic acid fermentation. This one fermentation without O2. That's why for the glycolysis in alcohol fermentation and glycolysis in lactic acid fermentation, for that glycolysis going on and on and on to continue, they need NAD+. They need continuous supply of NAD+. In cellular respiration, the electron transport chain is working. That's why produced NADH can transfer the electron into the electron transport chain. After giving up electron, it turns back into NAD+, and it can supply the NAD+ in glycolysis to going on. However, this one without oxygen, electron transport chain is blocking up. It is shut down. That's why for the continuous supply of NAD+ form in alcohol fermentation and lactic acid fermentation, NAD+ through the glycolysis, it is reduced into NADH. This NADH has to used up, means this NADH giving up electron, then it turns back into NAD+. So in alcohol fermentation, this NADH giving the electron to acetaldehyde and back into NAD+. In lactic acid fermentation, that reduced form of NADH giving the electron to pyruvate, producing lactic acid, lactate. After giving up electron, it turns back into NAD+. That NAD+ can be used up for glycolysis going on. That's why, because this one anaerobic process, that's why those produced NADH by glycolysis, they giving electron to somewhere else other than electron transport chain, then turn back into NAD+. So that's why we can see in alcohol fermentation and lactic acid fermentation with different types of fermentation without O2. As I mentioned earlier, this one showing just specifically for alcohol fermentation. So with the normal glycolysis going on, then by substrate level phosphorylation, they produce 2 ATP. Actually, that's the net productivity, 2 ATP. So first they consume 2 ATP, but later on they produce 4 ATP, so 4 minus 2. So net productivity of ATP, that's the 2 ATP by glycolysis. With this glycolysis going on, they need continuous supply of NAD plus form, as I mentioned. So this NAD can be used up in glycolysis and they are reduced into NADH. So without O2, without electron transport chain operation, that NADH, they turn back into NAD+. How? In this case, the pyruvate, that's the end product of glycolysis, it is broken one more step. So releasing out CO2 and turn into 2 carbon molecule acetaldehyde. So this NADH, they giving up electron to acetaldehyde. Then NADH turn back into NAD+, and it can be used up for glycolysis going on. Then acetaldehyde, after gaining electron from NADH, it is reduced into ethanol. That's why end product, that's the ethanol, ethyl alcohol. That's why we call this one alcohol fermentation. Lactic acid, almost same. So they making cycle between NAD plus form and NADH reduced form. And in this case of lactic acid fermentation, that NADH, they giving electron to the end product of glycolysis directly. Then pyruvate turn into lactic acid, lactate, and after giving up electron, NADH oxidized form into NAD plus form. So those NAD plus form can be supplied into glycolysis, and same thing. By the substrate level phosphorylation, only in glycolysis, net productivity of ATP, there's only 2 ATP. I mean, in glycolysis, that's all for the ATP production in lactic acid fermentation. In lactic acid fermentation, pyruvate is reduced by NADH, gaining electron from NADH, forming lactate as an end product with no release of CO2. Lactic acid fermentation by some fungi and bacteria is used to make cheese and yogurt and soy sauce and kimchi, like that. A human muscle cell uses lactic acid fermentation to generate ATP when O2 is not available. So when we go through very hard exercise, we can feel some muscle sore or pain like this, yeah, because of the accumulation of lactic acid in muscle cell.